So yeah, my talk picks up where Gabriella's left off. So as she mentioned, we're going to be making this annotation here available hopefully around the middle of November. And my talk is about how to access, the main ways to access and interact with this annotation. And the two main ways are through our website, ensemble.org, and through the companion FTP site. So the main part of our website is the genome browser, and as I'm sure you can expect, you can select the tracks of interest. So the ones I've chosen here are a gene track, which is showing sort of collapsed view here of the transcript, so you can see the exons. This sort of angle bracket is indicating the direction of transcription. There's also a track here of variants from a study from EVA, and as you can, and then just below here is our regulatory annotation. So you can see there's a legend down below, and we have red for promoters, as you can see lining up with the transcription start site as expected by design. And we also have the sort of yellow potential enhancers, and we have these gray sort of unclassified open chromatin regions. It's also possible to select the sort of tissue-specific tracks of interest to see the primary data. So here we have brain and embryos stage, and you can see the different histone marks that have previously been mentioned, ATAC-seq, which is open chromatin, and these different marks that line up here down below. And I think just one thing to highlight, one of the goals of AQUA FAANG is to help focus where variants might be of interest. You can certainly see that some of these variants are going to potentially land in regulatory regions which are going to be perhaps of much more interest than the ones falling outside. This is what the sort of interface looks like in practice. This is the region in detail view where you can see the tracks and the genome view here. The previous image I showed was exported from this export image button, which is useful if you want high-resolution PNG or a PDF. And then you can choose the tracks here with this configure this page. So when you choose configure this page, just to walk you through this a little practically, you get the different sort of sections for the different tracks. The one I'm talking about today is in regulation, and we have the regulatory build annotation track, but then this is where you would choose your sort of cell-specific primary data of interest. So we have brain. I'm selecting brain here just for example, here within the CARP. And we have, here's the cell tissue tab. We would then go to the experiment tab here. And this is where you can choose the primary data that's available for the cells that you've selected. So it's not all available, but just the ones that you've actually selected. We have categories of histone, open chromatin, transcription factors. And then after selecting our tracks, we then can sort of configure what we want to see from peaks and signals, as well as the epigenome activity that sort of Gabriella highlighted in terms of a summary of which of the tracks the regulatory features are active in. And this is sort of what it will look like. I just wanted to highlight a few features here just sort of to help with interpretation. So I think we'll start maybe here with the promoter. So we have a nice clear promoter at the start of this gene. And you'll see piling up sort of below are these marks, H3K4 trimethylation, H3K27 acetylation, which are very characteristic of an active enhancer. So there's very good evidence that you then have this promoter is active in these tissues. However, if you look across here, for example, at this unclassified open chromatin, the same sort of thing is occurring. We see a lot of sort of pileup of the same sort of marks. And I think, and similarly to some degree with these ones over here. And I think for there it's just worth highlighting that sort of the 5' ends of transcripts is somewhat uncertain, and where exactly transcription starts is not a totally definite thing. And so you might want to sort of consider this as functioning like a promoter. So don't take the classification as without needing interpretation. It's a guide to help you find things of interest, and then looking at the primary data, you can help understand what exactly it's doing in the tissues of interest. I also wanted to highlight we have sort of separate male and female for each of these stages. We could have combined these, but we've kept them separate because, as you can see, the data is very good and gives us pretty clear tracks, even with the biological replicates that are available. And you might be able to find sex-specific differences, but I think probably what mostly will be most useful is you can see how reproducible these tracks are. And that can give you a good indication that in this particular locus, the data is really coming through strongly and can be more reliable than one where you're seeing quite a fair degree of differences. You can also, given a specific feature of interest, you can click on it, and by following this ID link, you can come to this page. And I think the most important thing on this page here is the summary of regulatory activity below. And here you can see, for example, that this enhancer that we've selected on has been detected very clearly in the brain and has not been found to be active in the remaining tissues. And so before moving on, I just wanted to pause to point out things that I'm sure you're probably all aware of, but it's worth bearing in mind that our regulatory annotation is based on peaks, and peaks are based on statistical significance. And of course, if you have a little bit of a noise or assembly is not, problems, not mapping as well, you're going to get things not coming through as detected. They're not going to come through as active. So if something's inactive, it doesn't mean evidence of inactivity. So bear in mind that these false negatives are going to be a feature when it comes to looking at activity in this context. Also, in general, of course, if you have a more broad distributions of sort of the readout, whatever it is you're measuring, the activity, even if you have two classes, if they're overlapping, you're not going to find a clear distinction between these sort of on and off. So bear this in mind, but I'm hoping that you will find that this can still guide you into areas of interest for further exploration. Okay, so that's sort of the website part. We also do have the files available on FTP, at least these will be available at the time of the release. These could be where you want to go if you want to do some of the more detailed analysis, so it's worth just highlighting what's there. So we have regulatory annotation in a standard GFF format, and we also have the activity measurements in a tab-separated value file. And then we have the primary tracks, the big bed for the peaks, the big wig for the signals. So this will all be freely available and accessible. This is, for those who don't know, what a GFF file can look like. So this is some of the features, for example, with turbit. We'll have the chromosome here, the sort of coordinates on the chromosome, the type of feature, and so on. In this case, the 75,000 features that we are putting out there. The activity is the same idea in terms of one row for every feature, but we also have active or inactive sort of classification for each of the different tissues, cell types that we have. Now, I could just stop there, but I thought just to sort of whet your appetite, I'll do some really simple analysis just to show some of the patterns that are in the data already, perhaps to encourage further investigation. So, for example, taking the turbit, looking at the activity, we can sort of group it into brain, the different sort of tissues, so not considering embryo stages here. Sort of the late and early stages, we've got female, male. And now if you think of these as sort of ones or zeros, you can sum up for each tissue. So brain, for example, here on the first feature will be 4, and then 0 on the other tissues. As you can see, there'd probably be 3s and 2s and 1s. But if we filter just for the 4s and 0s, we're effectively finding those where there is very clear tissue-specific expression. So we're ignoring sex-specific differences, we're ignoring stage differences. But what do we see? Well, we can drop down from 75,000 to 20,000, so definitely more refined. And then if we look at how these sort of sets come out, so we're not picking up very many non-sex-specific differences in the gonad, which is perhaps not a huge surprise. And when these features are considered active, we find that they're active in the other tissues as well. And then we have sort of quite a lot of brain ones, although it's just over 5,000, so it's a fairly tractable reduction in the potential regions of interest in the genome. And so if you're looking at it from a whole genome point of view, this can then focus you down to some of the potentially most interesting regions. And so for illustration, I selected one of these random from chromosome 12. It's this particular enhancer here. And as would be expected, it's active in brain. This is how it was chosen. The bottom track is liver, and so it's not active in liver. Now, if we look at the actual gene that it's falling within, it is related to brain, at least by homology. And so this gives you some indication that probably you're finding something of interest there. And in fact, if we look at this sort of cluster of regulatory features, every single one of them is coming through as sort of brain-specific and not occurring in other tissues. But I want to also show you some things that are not quite as clear-cut. So for example, just starting on the right here, this regulatory feature is coming through as just brain, female, 18 months. But if you look below, you'll see there is some signal here, and the background, as you can see, is pretty good in terms of the regulatory features are really standing out nicely here. So there is some signal, but it's just not significant enough. And the question is, well, is this just one of those borderline cases of activity? As I say, you can't just take this as the label says. One needs to do further investigation. This one here, open chromatin, is same idea with brain. Perhaps there's some signal here, but I also just want to show it comes out with sort of late-stage embryo, which is again quite nicely suggestive that perhaps it's involved in sort of, or at least it occurs in the late stage of the embryo in terms of brain development potentially. So here are some of the things with fairly simple analysis you can already do with the annotation. And yeah, sorry, just to point out in Ensemble, we also have the individual transcripts. So if you, for example, have the promoter here of interest, you can see it's sort of at the start of this cluster of transcripts. There's also an open chromatin here, which actually has H3K4 trimethylation, H3K27 acetylation, the marks of a promoter, and it is near this transcription start site. So although it's not called as a promoter, I think there's a good case that this could be a promoter where you're seeing alternative transcription start sites. And here's the enhancer that we previously spoke about. Okay, and then really just because I didn't want the embryos to be left out, I chose some of the embryo active and inactive. We don't have an obvious way here of filtering out a lot of these false negatives. Some of the situations I've spoken about. So take this result with a pinch of salt, but it's nevertheless, at least for salmon, shows quite interesting trend in that we can see the activity increasing in terms of what we measure as the stages proceed. And at least on the left-hand side here, when something has been detected as active, and this could be a detection factor rather than necessarily biological, but when we are detecting it here, we see that it is also being detected through the later stages. These are probably not, the order here is probably not relevant. But that's, I think, primarily what I wanted to show in terms of what we have available, just, I think, to hopefully highlight that this annotation, the availability of this data, all of this really opens up a lot in terms of further research, and I think the quality of the data is really coming through clearly in that none of what we have been able to do would be possible without, really good data. And so we do have help and documentation, but please feel free to contact us, ask any questions. We're around during this conference, so please talk to us if you would like to understand anything in more detail. Acknowledgements again, same team that Gabriella mentioned. It's been a pleasure working with this team in developing this annotation pipeline, and thanks for the funders who make this possible. Thank you very much.